Raphael, I want to move on to our audience Q&A. We've had some really thoughtful questions come in. And so first up, I want to ask Raphael, this one of you. What are the challenges you are experiencing when sourcing allogeneic cells for cell therapies? Donor characteristics, collection methods, donor variability, and scalability.
Yeah, this is part of the curveball that people throw at us systematically and saying, hey, how are you going to be able to deal with all these issues when you use a healthy donor, or if you use even cord blood, uh, this is going to be kind of a limiting factor. First, do we have variability between donors? Of course. Even one donor will show variability depending on what he has eaten the night before, how many drinks he had, or whatever other things that happen in his life. So that's the reality of it. By definition, human beings are all different. But there are ways of identifying some specific criteria that we will consider are the one that will help us saying or stating this donor has good cells or better cells than this other one, based on a set of different criteria that we're going to be identifying. My team has done an extensive work already in vitro and analyzing different donors, and we're going to continue to do so to try to find those predictive markers that are going to tell us that if the donor cells have these type of different markers, they are more likely to have a better outcome in terms of how they're going to be behaving once they're engineered versus other type of donors. All this is basic science that everybody can do and knows how to do, and it's important to do it. I actually realize that many biotechs and academic centers are working extensively on these, working with blood banks, working with different stakeholders to try to provide answers and directions that are helping us better identifying what are the best donors and what is the best process from the time of harvesting of the cells to our time of engineering. All these steps are critical. And I always use a very simple rule of thumb with my team is that the timing and the number of steps has to be to the minimum possible to increase the chance of success. If you have a process that had too many steps and too many variables, you're setting yourself up for failure or for challenges. So always try to go on the most simple approach and then see how good you are with that simple approach. And then you just enhance and expand and improve if you think it can make a difference. And so this is one of the, I want to hope that the next couple of years we will see plethora of data showing how important donor selection is, but also showing how we can identify the good versus the not so good donors to improve our process and get better cells at the end. One last point I will say, as a big differentiator between an autologous technology and an allogenic technology, if we select the best cells and we engineer them, we are engineering the best cells, the healthiest one, the strongest one. So we are enhancing cells that are already good. When you have a patient that has gone through 5, 7, 10 different rounds of chemo or cancer treatments, the immune cells that you are taking from that patient to engineer them are really severely beaten up. And yes, you can engineer them and try to enhance them, but if they're already beaten up, they're already not at their best. And engineering them is going to be just engineering, not the best cells. That's why I strongly believe that there is an alternative with allogenic technology. By using the top level cells, engineering them and delivering them to patients, we can reach the efficacy that is needed to kill cancer cells.
Thanks, Raphael. Next up, I have a question for Tracy. What qualifies a patient to be a safe recipient for NK cells?
So for us, safety with the patient is paramount. Because we want to make sure that we're not harming the patient further, But we also have to be very careful in this palliative care model not to take a patient that isn't in a positive enough health state to where they're going to go and they're going to travel, and the travel is going to take a toll on them, and then they're going to get to Cancun where we're doing these infusions, and something negative is going to happen to them that doesn't have anything to do with our therapy. Well, we're still going to have to report it as a negative side effect because they were there receiving our treatment. So the first thing that we look at is does this patient have a terminal or recurrent disease? Now, with the palliative care construct in Mexico, if the patient is diagnosed with cancer and they want to avoid conventional therapy, that in itself will qualify them as a cancer patient to receive treatment. But we're really trying to go after the worst of the worst right now, which will help us with not only our safety data when we're going through the IND process and the overall clinical trial process, but it will help us to understand in this early stage what type of dosing is going to be effective for these kinds of patients. So the patients that we're accepting into the program currently are still pretty healthy. Yes, they've had some chemo. Yes, some of them have had some radiation. They're still able to walk, talk, ambulate by themselves, take care of themselves. Their liver function is in a good standing. Their overall organ function as a whole is in good standing. Their counts haven't been completely wiped out. And they're able to travel without the travel itself taking some sort of incredible toll on their body. So we are very specific in that, but we have had some patients that are still quite sick as far as where they are in their journey. So finding that balance is really integral to us, making sure that we make this available to patients that we believe we can truly help, but also not doing it at a risk factor that could be something that could jeopardize the company down the road when it comes to reporting the outcomes of these patient studies that we're doing. So looking at the overall patient wellness and doing a really rigorous enrollment process is something that we take very seriously. We're doing a full medical record review with our clinicians. We're doing multiple Zoom calls. We bring in the clinicians from Cancun as well to take a look at the medical records. And there's a multi-pronged approval process before these patients are ever seen on day one. They go through rigorous blood testing that has to be done a month prior to their infusion. Oftentimes it's 2 weeks prior to their infusion if there are some markers that we're looking at that maybe otherwise had been elevated in the past that we're really keeping a close eye on. And then once they're there, they're getting additional blood work prior to getting the infusion the day before that's being analyzed by both the Cancun team and our team. And then they're also getting blood work after that first infusion and then subsequent blood work that continues 2 weeks after and then every 30 days to ensure that they can qualify for the following infusions. So the process is quite intense, but it is one that we have gotten very comfortable with and one that we think sets us up not only for the best possible outcome for our patients, but also protects the company at the same time.
Thank you, Tracy. One quick question for Raphael. Freezing and recovering NK cells, is it a challenge?
No immune cells likes to be frozen. So it's a challenge for every. Yes, T cells are a little bit easier than NK cells. NK cells have a reputation of not doing well when you freeze them. All that is true, but it is also proven wrong every time we find a new way of doing that process and improving it. So as I saw, you probably, this is a theme I always say, if we just get stuck on technological issues, we will do nothing. You can freeze NK cell. NK cell don't like to be manipulated. They don't like to have a complex process. They can shut down. Yes, all this is true. Freezing them can make them less active than what they are if they are fresh. That is why many people are still only using fresh material, but it is feasible. And remember what I said earlier, it's a balance. You know, we had an interesting experience where our cells, when we were engineering them and transducing them, we were having extremely high results in transduction. We were in the 90+% transduction rate and having our cells super strong, super active. We ended up realizing that it's too much. And it's actually better, and it's known by the NK specialists will know what I'm saying, that actually having a 60 to 70% transduction rate is way enough to have good cells. So the freezing process is a little bit of the same. Yes, I can have a cell that is super duper on steroids strong and that will be crazy killing, but maybe 80% of that is good enough to be able to have an impact. And so there are challenges on the freezing and process on the cryopreservation, but the technology has evolved so dramatically in the past few years that I actually don't see this as a hurdle. It's more like what is the best piece that we're going to take and what is the best technology that we're going to take to be fit for purpose of our cells and make this work. But I don't see this as a stopping challenge. It's just a bump on the road that we will go around, like the people did back in the days with T cells, and like we're going to do with other immune cells when we're going to freeze them, no matter what.